So now that you know the basics of how RecA assembles and how RecA promotes strand exchange on model DNA substrates, let's focus in on how RecA carries out the strand exchange reaction in the context of the double-strand break repair pathway. There are two nearly identical duplex DNA molecules. One of them experienced a double-strand break. That double-strand break was processed by the RecBCD enzyme to give regions of single-stranded DNA, and those regions of single-stranded DNA end in 3' ends in the middle. So they're called 3' single-stranded tails. So that's exactly the type of DNA that RecA likes to assemble on. Remember, RecA assembles in the 3' direction. So if RecA monomers are getting on somewhere here, they'll continue to form a filament out to the end of this single-stranded 3' end. The same will happen on the other side. RecA will coat that region of single-stranded DNA. And then RecA is ready to do the strand invasion reaction. So the RecA filament has prepared the DNA for looking for regions of complementary sequence. So these regions of DNA can invade the other non-broken duplex and start looking for regions of complementarity. It does this amazingly fast, actually, that it'll sample different places all along the DNA, and when it finds a region that is complementary to its own sequence, it'll start forming the base pairs and the DNA double helix. And so this intermediate is the strand exchange intermediate, where the first strand has invaded. There's a second consequence of this invasion is that strand invasion displaces the complementary strand of the invaded duplex. And that can go ahead and pair with its complementary strand on the other broken duplex. So now we have pairing here. So now we have a molecule that has two strand invasion reactions that have been catalyzed by the RecA protein. The next thing that has to happen is that we need to repair the DNA that RecBCD degraded. So as you remember, RecBCD leaves the 3' single-stranded DNA. So that means there's going to be a 3' end here and a 3' end here. And 3' ends are great for being primers for DNA synthesis. So the part of the DNA molecule that had been degraded by the RecBCD enzyme can now be resynthesized by a DNA polymerase. The DNA polymerase that usually does this reaction in E. coli is DNA polymerase 3 holoenzyme, because remember, the RecBCD enzyme is probably degrading hundreds and even maybe in the thousands of nucleotides when it makes those long single-stranded tails. So we need a pretty fast, pretty processive DNA polymerase to carry out this reaction. So those ends destroyed by the 3' end processing by the RecBCD enzyme are now resynthesized using the unbroken DNA molecule as the template. So then the final intermediate of this whole reaction is an intermediate that looks like this, where you have two holiday junctions. You have this strand invaded here, and this strand invaded here. I know it doesn't look the same as the similar type of invasion, but if we flip the DNA molecules over a different way, you could have the cross be here. And we have new synthesis of DNA to replace that was lost by degradation by RecBCD. And now to finish the double-strand break repair pathway, we have to learn about two more reactions. The reactions that move these DNA branches, called branch migration, and the enzymes that cleave these DNA branches, which are called the resolvases, which now finish the recombination to give you two separated duplex DNA molecules that are somewhat altered from what they started with, but are completely intact. Now let's hone inside the RecA filament and look at how this strand exchange reaction might really be happening. I've already told you that RecA likes to assemble on single-stranded DNA, so there's clearly a single-stranded DNA binding site within the RecA filament. And here we're looking top down at the filament, so the filament's going around and around like this, and we can see the single-stranded DNA binding site right here. Now, there's also an empty site that can be filled by DNA, and that's the duplex DNA binding site. So as RecA starts the homology search reaction, it'll test different DNAs to see if they are complementary to the strand that's in the single-stranded DNA binding site. Many places will not be a match, and RecA will dissociate from those regions. But if RecA finds a match, it starts really exchanging the strands. So here we have 3 strands bound in the RecA filament, and the original Watson-Crick base pairs between the starting duplex are intact. But when strand exchange occurs, because RecA found a region, a complementary strand to itself, now the base pairs are going to switch. So these 2 strands are base paired, and this strand is kicked out of the filament. So in this manner, the RecA protein really catalyzes the exchange of one DNA strand for another between these 2 DNA molecules.